Good morning. Welcome to Provider Grand Rounds. We're happy you could join us today. We're very excited for this presentation this morning. So now I will turn it over to Dr. Hedstrom, who will introduce our speaker today. And just a quick note, the planners have no disclosures to share with the audience today. Thank you.
I have the honor to introduce Dr. Donna Deno as our speaker today. As we know, she's a professor here in the Division of General Pediatrics and also has a joint appointment in the Department of Global Health and an adjunct appointment in the Department of Health Systems and Population Health in the School of Public Health, and teaches the Global Maternal Child Health course. Her clinical work here at the University of Washington is at the Kent Des Moines Clinic, and her research interest includes childhood intestinal infections and enteric dysfunctions, which we'll hear about more today, both domestically and globally, and health services approaches to improve utilization and quality of health services in low-resource settings. She has been faculty in Ghana at the Department of Child Health and has served as a consultant to the World Health Organization and to non-governmental organizations working in global child health. She earned her MD from the University of Michigan, did her pediatrics internship at Brown, and has been us ever since with her residency and MPH here at the University of Washington. She will share results today that were recently published in the American Journal of Clinical Nutrition from a consortium of studies that were aimed at elucidating the pathophysiology of environmental enteric dysfunction. Welcome, Dr. Deno.
Thank you. I am so excited to share with you today results from the Environmental Enteric Dysfunction Biopsy Consortium. I have no conflicts to disclose. Our learning objectives today will cover the scope and impact of environmental enteric dysfunction, or EED, its mucosal pathophysiology, and immune and infectious signals. Before I get into all of that, I thought I might take a minute and explain how it is that I got involved in EED research. Way back when I was a young pediatrician in the 1990s, working in Ghana, I cared for many children with acute illness who had underlying undernutrition in various forms. And their undernutrition was not only a risk factor for their recovery from their acute illness, but also, of course, had a profound impact on their long-term health, development, and well-being. And those kids really stuck with me, and it was something that I really wanted to do something about and work on. My research focused on diarrheal disease, which is still a leading killer of children globally, unbelievably. But then I began to realize that asymptomatic enteric infections and enteric dysfunction had a major role in child undernutrition. And that's how I came to work in this area of environmental enteric dysfunction. So what is EED? Well, it's an asymptomatic disorder of the small bowel. Virtually all children in low and middle income country settings likely have at least some degree of EED, and it contributes to malnutrition in millions of children globally. It's thought to be acquired probably in the second half of the first year of life, when the period of exclusive breastfeeding ends and children are taking in other fluids and foods, and they're often contaminated with enteric microbes. And also it's developmentally the period when children normally start developing mouthing behaviors and have lots of environmental exposures to their oral pharynx that are, contaminated. So that's probably why it's acquired generally in that period of time. It has a similar pathophysiology, not exactly the same, which we will get into, but similar to gluten-mediated celiac disease. But in EED, the exact trigger is yet unknown. Probably specific enteric pathogens, perturbed microbiota, and or specific nutrient deficiencies. Whatever the underlying causes, it is characterized by reduced gut surface area. So let me see here, get the pointer out here. So you can see right here, this is a normal comparison biopsy, and this is a biopsy from a child with EED, and I think you can easily see that in the normal biopsy we have long finger-like villi, versus a markedly reduced surface area in the EED biopsy. It's also characterized by intestinal inflammation, gut permeability, leading to translocation of microbes and microbial products from the gut into the systemic circulation, and all of this leading to systemic inflammation. And all of this has a profound impact on child growth, particularly linear growth. So let's talk about linear growth for a minute. Stunted linear growth, or stunting, is defined as having a length for age or height for age z-score of less than -2. For those of us who are used to percentiles in the United States, this is the equivalent of less than the 2.3rd percentile. And in low and middle income countries, 1 in 3 children have stunted linear growth. And on a population basis, this has it's an incredible risk for mortality, delayed neurocognitive development, reduced adult capacity, later in life metabolic syndrome, especially among kids who were stunted who then have excess calorie intake, and chronic non-communicable diseases like prediabetes, diabetes, which is not only increasing in the United States, but also globally. Okay, we do not have a treatment for EED. And unfortunately, recent well-designed water, sanitation, and hygiene trials that also included nutritional interventions have shown little or no impact on EED. And this is probably because if you have a high level of pathogen exposure, sort of the standard wash interventions that were implemented were insufficient to overcome that level of burden, that level of exposure burden. So you may not have heard of environmental enteric dysfunction before. Perhaps you've heard of tropical enteropathy. That was the old terminology, and then the terminology changed to environmental enteropathy to emphasize that the precipitant is environmental rather than geographic per se. So for example, EED has been seen in non-tropical Chile and among Aboriginal populations in Australia. And about 10 years ago, I was involved with a group that really advocated to the name change to environmental enteric dysfunction, to really drill down on the dysfunction component. What is going on with this disorder so we can identify therapeutic interventions? However, this disorder was first described in the 1960s, first among expatriates, military personnel, Peace Corps personnel residing in low-resource settings, and their enteropathy resolved upon repatriation to high-income settings. It was also seen in immigrants, mainly from Pakistan and India, and again, upon residence in high-income countries, their enteropathy resolved. And there was a flurry of interest in this topic in EED in the 60s and 70s, including the findings that associated EED with poor growth. But this was followed by a period of time where there was a dearth of investigation in the topic until about 15 years ago, when there was a resurgence of interest in the topic. But it was not concomitantly accompanied by investigation of the target tissue, the intestine. And you can see that from this Web of Science search, which certainly doesn't cover all of publications related to environmental enteric dysfunction, but you can see that in the orange, these are publications related to mucosal investigations versus the blue line are all publications. So why is this important? Well, a lack of understanding, first of all, we don't have a specific biomarker for EED. That's a problem. Next, a lack of understanding of the small bowel in EED is really a barrier to therapeutic targeting. And I would say this is also an equity issue. We have standards for, we would not tolerate a lack of understanding of the mucosa in disorders that are prevalent in high-income settings in the United States, like celiac disease and inflammatory bowel disease. And also, clinical standards in these disorders often require mucosal pathology for diagnosis and disease monitoring. And yet, this is not the case in low and middle income countries, where we have an enteropathy that is highly consequential and prevalent. Okay, so it is with this backdrop that in 2015, the Gates Foundation put out a call for proposals to elucidate the pathophysiology of EED through intestinal tissue investigation. Really, the idea behind this was to really start looking for therapeutic targets. And they awarded 3 studies, and you see the pictures and names of the PIs and their key co-Is in this slide. So the 3 studies that were awarded, starting with the Tropical Gastroenterology and Nutrition Unit in Zambia, Aga Khan University in Pakistan, and the International Center for Diarrheal Disease Research, or ICDDRB, in Bangladesh. The latter 2 of these proposals also partnered with U.S. centers in Cincinnati and Charlottesville, Virginia, to recruit children as comparison groups. Okay, so let me give you a little bit of an overview of the EED enrolling centers, starting first with the 3 EED enrolling centers. Their approach was to screen children in the community, going door to door, using screening anthropometry to identify children with undernutrition. And I'll tell you a little bit more about how they defined undernutrition in a little bit. But upon identifying kids who were undernourished, they then provided nutritional supplement. And for the children who did not respond to the nutritional supplementation, they underwent a medical workup to identify causes of their poor growth. And for those children for whom a medical condition was not identified, they were invited to endoscopy and duodenal biopsy, and extraintestinal specimens, blood, urine, stool, were also collected for research purposes. Now, for those of us who do failure to thrive workups, this probably looks very familiar. But these were really the first intestinal biopsy research studies done on EED in modern times, and really the first intestinal studies on EED in decades. And they represent the largest collection of EED biopsies to date. Okay, so drilling down a little bit on the individual studies, in Bangladesh, they recruited children based on length for age Z-score. In Zambia, they recruited children based on wasting, or weight for length, low weight for length Z-score, or thinness, but most of the children were actually recruited based on their linear stunting status. In Pakistan, they recruited children based on wasting, although most of those children also had stunted linear growth, which is not surprising. The supplementation included varied from daily eggs, milk, micronutrient supplements. In Zambia, they used a high-energy protein supplement, and in Pakistan, they used a ready-to-use supplementary food. The duration of the treatment lasted for anywhere from 2 months to 4 months, depending on the type of intervention. And you can see here that the age at supplementation was quite similar across the 3 studies. Now, in our US comparison cohort, they recruited children who were presenting for endoscopy and biopsy for clinical reasons, like, for example, for abdominal pain. And for the purposes of the analyses that I'm going to present today, we parsed these kids out into 2 groups. First was the group of children that had no medical diagnosis consistent with an esophageal, gastric, or intestinal disorder, and their biopsies were read as normal by the clinical pathologist. And the second group were newly diagnosed, were children with newly diagnosed celiac disease. Okay, so these studies were awarded in 2015, and then in 2016, about a year later, the funder, the Gates Foundation, decided to bring these studies under the umbrella of a consortium. And this was because of the quite novel studies, the populations engaged in the research, and really the desire to glean the most from the precious samples that were being collected. So the consortium was formed as a shared scientific platform and to harmonize procedures to the extent possible. Now remember, these studies had already been funded, and they had already developed protocols and were starting to screen children in the community. So we did try to harmonize things to the extent possible at that point. We also developed a centralized and secure telepathology platform, and the consortium, I think one of the main goals was to extend opportunities for advanced tissue analytics. These were things that weren't planned as part of the individual original studies. So, for example, developing and validating an EED histology scoring system for diagnosis and assessing disease severity, and I'll be telling you a lot more about this in a few minutes. We developed an immunohistochemistry platform to further interrogate these tissues for to better understand pathophysiology. Due to time, I won't be going over the quantitative mucosal morphometry assessment, but if you have questions about that, I'd be happy to address those. And then the consortium really also allowed us to leverage power through enhanced sample size. We were able to really conduct analyses that wouldn't have been possible with the individual sites alone, and we were able to explore differences between the EED sites. So I had the incredible privilege of being one of the co-coordinating investigators in the consortium, along with Drs. Philip Tarr and Peter Sullivan, who are pediatric gastroenterologists. In the consortium, we were able to recruit 5 terrific gastrointestinal pathologists. These pathologists developed the scoring system. They scored all of the biopsies. They developed the immunohistochemistry platform and really helped with the interpretation of the histology and immunohistochemistry results. We had our consortium coordinating body, and that including for the telepathology platform. And we had a terrific set of data analysts led by Dr. Kelly Van Buskirk and our really fantastic epidemiology PhD students. I think David and Brooks are online, and Marwa is here with us in the audience today. And we also had the incredible privilege of working with Monica Mwita, who's a PhD student who resides in Zambia, and she was an awesome part of our consortium analyst team. Okay, this all culminated in the dissemination of the results recently as a special supplement to the American Journal of Clinical Nutrition, where we published 8 papers, and I'll be giving you some highlights of just a few of the papers. But before we get into that, I really want to acknowledge the tremendous effort that the PIs and the research teams put in on the ground to make these studies possible, starting with community outreach and sensitization, community-based anthropometric screening of over 11,000 children, enrollment in nutritional intervention of over 1,500 children with weekly or daily, most of the time it was daily follow-up and response assessment, sample collection and processing, and medical workup of about 1,000 children who did not respond to the nutritional intervention, data collection and management, and of course, ensuring safe procedures in the endoscopy suite. So there were 449 children that underwent endoscopy, including 301 in our EED endemic settings, and there were no serious adverse study events. So I'm going to provide you with some highlights from the consortium, starting with our enrollment flowchart, which looks busy, but I will just point out some highlights. So let's just start with the 3 EED recruiting centers. Half of the children who completed the full anthropometric screening process met undernutrition criteria for the studies. Half of the children met the undernutrition criteria. And of those, 1,537, or 83%, consented to the nutritional supplementation. Of those who completed the nutritional intervention, only 198, only 15%, had an anthropometric response to that nutritional intervention. Of those who completed but did not respond, notably only 4 or 0.3% had lab evidence suggesting celiac disease, and only 68 or 6% had an identifiable medical cause of their growth faltering. So of the approximately 1,000 who were eligible for endoscopy, 52% declined to consent to the procedure. So in the end, we had 301 children from the EED recruiting sites who were endoscoped, and of those, 291 had at least one histologic slide of sufficient quality for assessment. And you can see the distribution here across the 3 EED centers. Now, on the US comparison side, we had 148 children who were presenting for endoscopy and biopsy for clinical indications, and who consented to be in the study. 82 of them, or 55%, were excluded because they had histologic evidence that could be consistent with an esophageal, gastric, or intestinal pathology other than celiac disease. So then we were left with 22 children with newly diagnosed celiac disease and 44 children in the what we call the normal comparison group. Okay, so looking at some demographic characteristics across these cohorts, first of all, it's not surprising that the children in the US comparison group were older. They were being recruited because they were presenting for endoscopy for things like abdominal pain. Next, if we look at some of the anthropometric data here, maybe zeroing in on the length for age Z-score, we see that in the EED enrolling centers, the median LAZ neared -3. -3 is the cut point for what is considered to be severe stunting, and that would be equivalent to less than the first percentile. And then we see that in the US comparison groups, that they really did not have anthropometric, they didn't have undernutrition by anthropometry. Okay, so let's talk about what we learned from these biopsies. Perhaps the most prominent contribution of the consortium was the development and validation of an EED histology scoring system to identify EED and grade its severity, as described in this paper that was led by Dr. Paul Kelly from Zambia. So to start off with, the consortium pathologists inducted an agnostic list of 11 histology parameters to represent the breadth of what the potential pathophysiology of EED could look like, kind of like a kitchen sink without bias to favored features. So these histology parameters would be familiar to pathologists and therefore could be transportable to other settings and other studies around the world. We had 571 biopsies from 357 children across the 5 sites that were paraffin embedded, sectioned, and stained. The histology slides were then scanned on site and uploaded to our central telepathology platform. And each of these 571 slide images were then blindly assigned to 2 or 3 of the 5 consortium gastrointestinal pathologists, and they were blinded to study center. And they scored these slide images across all of the 11 histology parameters, as well as 3 slide staining and preparation quality metrics. There was high inter-reader agreement between the pathologists, and the pathologists created an atlas that could guide uniform and objective application of the scoring system. This is a really terrific resource, and it's freely accessible and has been published as a supplement to the histology paper. Each of the 11 parameters could be scored on a scale of 0, which was considered normal, to 3 or 4, which was considered the most aberrant. So a low score is more normal and a higher score is more abnormal. And an example of the scoring system for villus architecture is shown on the right. Okay, so what did we find? Well, first, we had 3 parameters that were not prevalent in any of the cohorts. So we quickly jettisoned those and didn't really consider them much further. That left us with 8 histology parameters. And just a reminder that our primary analysis was to compare the EED group to the normal comparison group. And to So the first histologic parameter that was the most abnormal in the EED cohort was villus architecture. And to kind of oversimplify this a little bit, what this meant was that the normal villi were shortened or sometimes absent, meaning there was a reduced surface area for nutrient absorption. Then we also saw a paucity of Paneth cells and goblet cells. Paneth cells and goblet cells are derived from the crypt cells, from the intestinal crypts. And Paneth cells secrete from the stem cells in the intestinal crypts. Paneth cells secrete antimicrobial peptides, and goblet cells secrete mucin. So these are really important in protecting against infection. And I think an important question for future research is, why did we see a paucity of these cells? Was it because of poor stem cell differentiation, or were these cells being exhausted due to response to an exposure, a microbial exposure? We see that the Paneth cells and goblet cells, so the villus architecture was more abnormal in the celiac comparison group than in the EED group, although it was also very abnormal in the EED group. These two features, Paneth cells and goblet cells, were the only features that were more abnormal in the EED group than in the celiac group. Okay, next most abnormal feature was intraepithelial lymphocytosis, which is a hallmark feature of celiac disease, but was also highly abnormal in the EED group. And then we see intramucosal Brunner's glands. Okay, Brunner's glands normally reside in the submucosa. We did not have submucosal biopsies. We had mucosal biopsies. Brunner's glands secrete a mucin that contains like an alkaline fluid to protect the mucosa, the duodenal mucosa, against gastric acid. And their location in the duodenal mucosa is, to any large extent, is considered abnormal, and you see it in conditions like peptic duodenitis. Now, we didn't really see intramucosal Brunner's glands in EED, but they did help us distinguish from the normal tissue. So they were this feature was highly different in the EED group compared to the normal group. And then our last three parameters were also differed from the normal group, although not to as great of an extent as our top 5 features. Okay, so if we look at comparing the EED group to the normal comparison group, the figures noted in bold represent histologic parameters that were statistically significantly different between the EED group and the normal group. And the ones that are in orange were worse in the EED group compared to the normal group. And then again, we weren't seeking to develop a histology scoring system to differentiate EED from celiac disease. We already have good lab markers of celiac disease, and coupled with histology, that is how the diagnosis of celiac disease is made already. But we wanted to compare the pathophysiology between the two. And again, the bolded numbers represent those histology parameters that were significantly different. The only two parameters that were worse in the EED group compared to the celiac group, again, were the Paneth cell and goblet cell paucity, with Paneth cells being statistically significantly different in the EED group compared to the celiac group. Now, you may have noticed these pesky non-scorables, especially for villus atrophy, villus architecture, and Paneth cell features. And this was largely in the EED cohort, and it was due to technical issues that we had with slide staining and tissue preparation. And thanks to QI processes during the consortium that was led by the pathologist, we were really able to address these over time, and I really think these issues can be mitigated in future studies. So this is a representative, these are representative histology slide images from our consortium. So on the left here, we have a biopsy from the normal cohort, and then on the right, we have a biopsy from our EED cohort. And so what you'll notice on the left is we have, again, long finger-like villi, a nice extensive surface area for absorbing nutrients. Then you'll also notice these oval-shaped ghost-like cells. Those are goblet cells. And then down here in the crypts, we have these pink-stained, kind of granulated pink-stained cells. Those are Paneth cells. So moving over to our EED biopsy, we see that there's a greatly reduced surface area. Where there are villi, where we see the villi, they're quite shortened. We see a lesser number of goblet cells, and it's very hard to find any Paneth cells in this biopsy. And then the other thing that you might notice is this really extensive mononuclear inflammatory infiltrate in the lamina propria. So that's just to give you an idea from our representative slides. Okay, so we had all of these individual parameters, and that was to help us describe the histopathology of the disorder. But next, we really wanted to derive a summative score, a way to pull this all together that can be used in future applications. So we used test and validation sets to create receiver operating curves, or ROCs, and calculate area under the curve, or AUCs, to test summative score performance. So AUCs quantify the overall performance of a diagnostic test, especially when you're comparing between two groups, so in our case, EED compared to the normal cohort. And if you have a diagonal line, that basically represents random chance and corresponds to an AUC of 0.5. If you have a perpendicular lines, that represents a perfect performance and corresponds to an AUC of 1. And typically, an AUC of 0.85 is considered to be pretty good. So we tested various summative scores, and I'm just going to highlight 3 of them today. First, we started out by just simply summing all of the histology parameters. And this didn't really perform very well, and it doesn't take into account that some biopsies have non-scorable parameters, making a simple sum not really comparable between 2 different biopsies. So next, we tested a total score percent. Now, this variable accounts for those histology parameters that could not be scored. So the numerator is the sum of the scores of all the histology parameters that could be scored. The denominator is the maximum possible scores of those that were scored. So if all of the parameters were normal and they were scored to 0, the total score percent would be 0%. If they were all scored most abnormal, most highly aberrant, the total score percent would be 100%. Okay, and this performed really well. We had an AUC of 0.992. Next, we created a total score percent for those top 5 most informative histologic parameters to see if we could pare this down a little bit, make it a little bit more simple. So we called this the total score percent 5, or TSP5, and we did in this score inverse code those Brunner's glands that were more normal in the EED cohort. But they did help us distinguish between EED and those normal biopsies. So this TSP5 performed very well. It is very easy to calculate, and it's very transportable to other studies and other settings. What I came to learn through this process is that it's really highly unusual for such epidemiologic rigor to be applied to histology scoring systems. And I think that the development and validation of this summative high-performing histology score for future applications was really a major accomplishment of the consortium. So you can see from the distribution of the scores in the EED group compared to the normal group, there was really high separation between the groups. The TSP5 readily distinguishes EED. And as I said, we never set out to develop a scoring system to distinguish EED from celiac disease, but I do think it's interesting that despite the fact that most of the histology parameters were worse in celiac disease, our summative score, the TSP5, did identify children in the EED cohort as having a higher summative score compared to the celiac group. Okay. Next, I would like to tell you just a little bit high level of about our immunohistochemistry analysis, which aimed to identify gene expression patterns in EED. We utilized antibodies that were attached to chromogens, in other words, color, that bind to antigens of specific gene products on a biopsy section. Where possible, we used multiplexed antigen stain, meaning that we tried to assess for as many antigens as possible on a given biopsy in order to preserve the biopsies. So we started off with having a brainstorming session. What antigens would we like to see on these panels? And we came up with a list, and then the pathologists they tested the performance of the various antigens that we were interested in. And in the end, 13 targets performed reliably, and these were combined into 6 different panels. And each of the panels contained 1 to 6 antibody stains representing a whole range of immune and epithelial differentiation and function. And you see on the right here 3 sample panels. So participants who had sufficient tissue remaining after the histology assessments were included in the IHC analysis. Scanned IHC slides were digitally read. They were machine read and quantified. The distribution of the IHC markers was not normal, so they were log transformed prior to analysis and regression models. And what we found was there were 7 targets that were differentially expressed in the normal tissue compared to the EED tissue. So in EED, we saw, you'll see in the black font, different immune cell product patterns. And then we also, in the orange font, found a reduction in the brush border. And then in the pink purple font, you see Reg1b, which is a marker of epithelial repair following injury. So due to time, I'm not going to go into these in detail, except for I'll tell you a little bit about one of them, lipocalin-2. So lipocalin-2 has many functions, including innate immunity. This was the most highly upregulated marker that we saw in the IHC analysis. It was 659% more highly expressed in the EED biopsies compared to the normal biopsies. Fecal lipocalin-2 has been shown to be associated with celiac disease histopathology, so we think this is a promising biomarker to be investigated in the future as a potential biomarker for EED. So to summarize our IHC data, we saw specific epithelial and immune responses to an environmental trigger, probably microbial, and we found markers that we think support future biomarker validation, not only for lipocalin, but also for Reg1b. Okay, and I'll just provide some really high-level results from our transcriptomics analysis, which was led by Dr. Chelsea Marie, who trained at UW and is now at the University of Virginia in Charlottesville. The aim of this investigation was to identify dysregulated genes and pathways in EED using biopsy-generated RNA sequencing libraries. So these were biopsies from the children that were then where RNA sequencing libraries were extracted. Okay, we basically the first 30 to 50 children from the EED cohorts had biopsies that were designated for RNA-seq, and the majority of the comparison group had sufficient biopsies for RNA-seq. And we batched these RNA sequencing libraries, and we applied 3 statistical methods. One, first was differential expression analysis to look at the differences between the genes in the cohorts, and then gene set enrichment analysis to interpret the function of the identified genes, and lastly, weighted gene network analysis to identify genes that are related to each other within the EED cohort. So to understand how these genes are co-regulated within EED. So what we found, this volcano plot shows upregulated genes on the right, downregulated genes on the left. Those that were statistically significantly elevated are in orange. The blue are statistically significantly downregulated. Obviously, there were many, and we don't have time to go into all of them, but just to say that the most downregulated genes represent lipid metabolism and mucin encoding genes. And then on the upregulated side, the most highly upregulated genes included DUOX2 and its chaperone DUOX2A, and these encode NADPH oxidase, which is implicated in small bowel epithelial antimicrobial response. We also see FDCSP, which is involved in antibody response and mucosal IgA production, and our friend lipocalin-2, again involved in innate immunity. So these various analyses really helped us corroborate data across the different methodologies. So applying the weighted gene network analysis within the EED cohort. So this was to look at what genes are co-regulated with lipocalin-2 among the children with EED. And we found that it was mainly the genes that were co-regulated were other antimicrobial genes. And the other thing we found was that lipocalin-2 was the most highly correlated gene with the EED histology, including our summative TSP5, the goblet cell paucity, and villus blunting. And lastly, the functional pathway analysis. This is comparing the children with EED to the normal cohort, trying to understand what the meaning of these genes are from a functional pathway perspective. We found disproportionate representation of immune pathways and processes across diverse functional categories. And we think that these are likely in response to a microbial trigger. So the last paper that I'll provide some highlights was led by Dr. Najeeha Iqbal from Aga Khan University in Pakistan, the aim of which was to identify enteric pathogens that could predict EED histology severity. So we used stool samples collected from children prior to their endoscopy to assess for enteric pathogens using, well, there were two different platforms that depended on which site related to the platform that was used. These were PCR platforms, and we applied multivariable linear regression to associate the pathogens with histology. So what we found, first of all, 84% of the children in the EED cohort had sufficient stool sample for this analysis. And what we found was that enteropathogen carriage was incredibly prevalent. And these are pathogens. I'm not talking about commensal E. coli here. These are bona fide pathogens, and these children did not have diarrhea at the time of the stool collection. We found that 97% of the stools had at least one pathogen, and these data converge with other large studies that have shown similar asymptomatic pathogen carriage results. We found a mean of 4 pathogens per stool in these kids. And our leading pathogen profile also converges with other large studies, with the exception of Giardia. I mean, we found Giardia at a higher prevalence than other studies have. So I'm just going to share with you one slide result, and this is from a restricted pathogen data set where we only examined pathogens that were detectable, that were available across all 3 centers. So this is not the breadth of what's represented in the paper, but from a high-level perspective, we found that Campylobacter and enterotoxigenic E. coli, which in the United States we think of this as being a cause of traveler's diarrhea, but is also considered pathogenic in low and middle income settings. These were associated with our summative score, the TSP5, and Campylobacter was also associated with intraepithelial lymphocytosis. Shigella and norovirus were associated with enterocyte injury. So even though this is just, I'm showing you, just one set of data here, I just want to make the point that we make a lot of assumptions about enteric pathogenesis based on mouse models with very few human data. I am not aware of any other study that has looked at enteric pathogen association with histology. And I think there's a lot to be gleaned from these types of studies. Of course, mouse models are important, but human data is also obviously very important. So in summary, I think it's important that I point out that there were many limitations to our study. One was we had diverse eligibility criteria and procedures, and our U.S. comparison cohort was of an older age. Despite these limitations, this is the largest EED small bowel mucosal study to date. We had unbiased histology assessments. We had digitalized quantitative immunohistochemistry readouts and unbiased transcriptomics analysis. And we were able to look at the relationship between readouts from non-intestinal specimens like stool, the pathogens in the stool, to histology. So in conclusion, we were able to develop this 5-feature histology index score, the TSP5, which is easy to calculate and identifies EED and EED severity and is easily transportable to other studies. Our histologic, IHC, transcriptomic, and pathogen data converge to suggest a microbially mediated environmental trigger for the pathology that we see in EED. And this really offers us opportunities to investigate therapeutic targets, and I think it also offers opportunities to further define the pathophysiology of the disorder, like, for example, with microbiome assessments, spatial transcriptomics. So I'd like to acknowledge our funder, the Gates Foundation, my fellow co-coordinating investigators, our consortium team, the consortium gastrointestinal pathologists, but I especially really want to point out the very hard work of the principal investigators and their teams at the ICDDRB, Aga Khan University, Tropical Gastroenterology Unit in Zambia, as well as at Cincinnati Children's and UVA, and of course, the families. If you happen to read any of the papers and you have any questions or comments, I would love to hear from you. And I thank you for your attention, and I welcome any questions or comments.
That was a very nice talk. One of the questions I have is there's a number of specific vitamins and minerals, et cetera, that can be that are absorbed from the gut and can be treated either by bypassing the gut and giving subcutaneous injections or giving extremely high levels of vitamins, say B12, that are not included in traditional nutritional supplements at that level. In EED, do you have any such important factors that could explain pathology in other organ systems?
Yeah, I think that's a really important question, and I think one of the reasons why we see nutritional interventions that haven't had the impact on EED that you would maybe expect is that the micronutrient supplementation, including what we used in this in the consortium studies, met the 100% RDA, which is likely highly insufficient to overcome the malabsorption. And so actually I'm working on a proposal right now to include an intervention that includes a much higher dosing of micronutrients to see if we can overcome that. But your point about the sub-Q application is well taken.
Thanks so much for this presentation. I'm curious, in terms of the pathogens that you identified, were there any geographical differences, or was it pretty uniform across all 3 study sites?
It's challenging, because in Zambia they use the Luminex platform, and then at AKU and in Bangladesh they used the TACMAN array cards. So we did a sub-analysis where we looked at the data from the 2 South Asian centers that use the same technology. And so first of all, yes, we did see some pathogen differences, which is not surprising, between the cohorts. But then we also did a sub-analysis where we just used the TACMAN array data, which actually gave us a much broader panel of pathogens to analyze, and we did find additional pathogens that were associated with histology, like enteropathogenic E. coli, which wasn't available on the Luminex platform. And we were also able to look more specifically at like atypical versus typical EPEC, and so yes, the answer to the question is yes. I don't think we had enough power, though, to look at the association with histology by site.
It's really incredible work, Donna. Thanks for sharing it. Yeah, I can imagine the breadth of the team and what was accomplished is really phenomenal. Can you talk a little bit about the hypotheses that your team had around the pathogens in particular, and their whether that would be some in addition to the therapeutic intervention of nutrition, like maybe that wasn't enough. I found it fascinating that the studies in the 60s, people who had EED and then came back to the US, that it seemed to resolve.
Yeah.
So maybe just curious on what your team thinks that the significance of those pathogens are to the process.
Yeah, well, we think that the combination of what we found in the pathogen analysis, but also the combination of what was found in the immunohistochemistry, histology, and transcriptomic analysis all point to EED being a response to a microbially mediated environment. And it's really unfortunate that those really well-designed water sanitation and hygiene intervention trials didn't show an impact on EED. That was a major disappointment. But I really do think it's because we probably are dealing with a level of contamination that those interventions were not able to address. So the question for interventions are, well, from a prevention standpoint, what can we do to further reduce the contamination, the microbial contamination? And then from a therapeutic standpoint, what can we do to address? So, for example, would intermittent azithromycin, for example, help reduce the burden of EED? Azithromycin has been shown in studies where it's been used on a community basis to reduce all-cause mortality and high mortality settings. This is among children. And it is recommended by the WHO in high mortality settings to be given, I believe it's every 6 months, to children. So the question is, could that have an impact on EED? And there's some, azithromycin not only has an antimicrobial response, but it also, properties, but it also has anti-inflammatory properties. So, there's a lot to learn from. I think we need to move forward now with interventional trials. I think the days of cohort trials in EED are over.
And we have a question online from Michael Arndt. Hi, Dr. Deno. Thank you for a great presentation and commitment to this important topic. I'm curious whether LCN2 has been linked to small intestinal bacterial overgrowth in this consortium studies or others. Also, are there any key findings from machine learning models applied to the transcriptomic data?
Yeah. Hey, Mike, thanks for the questions. So we didn't look at small bowel bacterial overgrowth in the consortium because those analyses were only done in the Bangladesh cohort. So in addition to the consortium data, all of the individual sites are publishing their own individual data as well, and many of them have other analyses that they've conducted that wasn't done across all 3 centers. So, they're publishing their individual data. As far as I know, I haven't seen LCN to investigated in association with SBPO, but that would be very interesting. And as far as machine learning approaches go, I really hope that that is a next step that can be utilized, not only for the transcriptomics data, but perhaps for the histology data as well. So hopefully that, and when the Pakistan group has actually been working on that. Thank you.